Hi everybody, welcome back. So today we're going to be finishing chapter 18, sections 3 and 4. So previously, remember we were discussing the electron transport chain, so how these electron carriers, NADH and FADH2, are then oxidized and transfer their electrons through the chain in order to reduce O2 and make H2O. All right, so this is sort of where we're at now. So what happened how is the transfer of these electrons used to develop ATP? Okay, so our learning objectives here, I'll pause a moment so that you can read, print, however you need to do. All right, let's continue on. So, as you recall, electrons are transported through the electron transport chain in a cascading of redox centers. All right, so NADH and FADH2 transfer their proteins to complex 1 or complex 2. Not their proteins, they're electrons. These are either complex 1 or complex 2. And then those electrons travel through the electron transport chain, eventually to reduce oxygen into water. But at the same time, as these electrons are going through these redox centers and releasing energy, this energy is used to pump protons across an electrochemical gradient. So we're taking protons from a low proton concentration matrix into a high proton concentration intermembrane space. Now, since we're going against the gradient, this is an endergonic process that requires energy in order to do this. So the energy that is released when these electrons are sent through these redox centers is actually what supplies the energy in order to pump these protons across. Then the protons are able to come back into the matrix in only one way, and that's through the protein complex ATPase. The energy that is released as these protons go from a high proton concentration area to a low proton concentration area is then what is harnessed to make ATP. So this concept is actually called the Chemiosmotic Theory, which was proposed in 1961 by Peter Mitchell. So just to kind of re-summarize, this theory states that the free energy of electron transport is conserved by pumping protons from the matrix to the intermembrane space in order to create an electrochemical proton gradient. The electrochemical potential of this gradient is what is harnessed in order to synthesize ATP. So there's 4 principles of the chemiosmotic theory. 1, oxidative phosphorylation requires an intact inner mitochondrial membrane, so no holes in that membrane space in which ions can freely go through. The inner mitochondrial membrane must be impermeable to ions whose free diffusion would discharge an electrochemical gradient. So that also encompasses sodium as well as potassium and magnesium. Because if these ions could also go through the membrane, then that would also dissipate the electrochemical gradient we're trying to make. Electron transport results in the pumping of protons out of the matrix into the intermembrane space, which thus creates the electrochemical gradient. Compounds that increase permeability of the inner mitochondrial membrane to protons will dissipate this electrochemical gradient. This then allows the electron transport chain to continue, however, would inhibit ATP synthase, thus uncoupling the electron transport from oxidative phosphorylation. So remember when we were talking about the case study with DNP, DNP was able to bind protons and transport it across the membrane back into the matrix, thus dissipating the proton gradient, thus getting rid of all the energy that we harnessed in order to drive ATP synthesis. So electron transport is what actually generates the proton gradient. So the export of protons from the mitochondrial matrix against the gradient is an endergonic process. Remember, we already talked about that. We require delta G, or we require energy in order to drive this process. The discharge of the gradient is exergonic. Okay, so we get a negative delta G as protons cross back into the matrix through ATPase. This free energy is harnessed by ATP synthase to drive phosphorylation of ADP. Phosphorylation of ADP to ATP requires about 40 to 50 kilojoules per mole. This amount of energy requires at least 2 protons re-entering the matrix in order to phosphorylate 1 ADP to make ATP. So we require no less than 2 protons to make ATP, 1 molecule. All right. So the protein complex, or the enzyme complex that governs this process, is called ATP synthase. It's also known as proton pumping ATP synthase or F1, F0 ATPase. It's a multi-subunit transmembrane protein that's about 450 kilodaltons in size. So it's a fairly large protein complex. F0 is a water-insoluble transmembrane protein with 8 different subunits. F1 is a water-soluble peripheral membrane protein that is comprised of 5 different subunits. And a lot of people say that it looks sort of like a lollipop. Let's take a little closer look. So the F1 component, which we said is this, the lollipop part of the protein complex, is comprised of 3 alpha, 3 beta, a gamma, a delta, and an epsilon subunit. The alpha and beta subunits are nearly identical, and they share sequence homogeneity. And they are arranged in an alternating fashion, sort of like people sort of think of it as like slices of an orange or segments of an orange, as you can see here. And they alternate, so you get alpha, beta, alpha, beta, alpha, beta around, and they sit on top of the gamma, the gamma subunit. The alpha and beta subunit arrangement gives pseudo threefold symmetry. So each alpha beta dimer is sort of arranged into 3 subunits. However, this protein is not actually symmetrical because each alpha beta pair adopts a different conformation, each with a different substrate affinity, and we'll talk about that in just a second. The FO component, which is that component that's in the membrane, consists of an A, B, and C subunit. So you have one A unit, this B2 unit, and then you have 2 Bs, 1 A, and then you have anywhere from 9 to 12 C units, depending on the species. The C subunit consists of 2 alpha helices that associate to form a ring, as we see here, that's embedded in the membrane. The epsilon subunit of F1 interfaces between the C ring and of FO and the gamma stem of F1 to create a foot-like interface. The delta of F1 creates an interface between the alpha beta dimers and the B helix. so that it is anchored to the highly hydrophobic A subunit. So ATP is synthesized by the interaction of both of these FO and F1 components. Translocation of protons is carried out by the FO, while catalysis of formation of the phosphoanhydride bond of ATP is carried out by F1. Coupling of the dissipation of the proton gradient with ATP synthesis requires interaction between the F1 and the FO. So this is an example. So ATP synthase is an example of a binding change mechanism. The alpha-beta subunits have 3 different conformational states. One that binds the substrates and products loosely, which is the L state. One that binds them tightly, which is this T state. And one that does not bind the products at all, the open state. The free energy released on proton translocation is harnessed to interconvert these 3 states. So let's look at it first. ADP and inorganic phosphate come and bind the L state. A free energy-driven conformational change converts the L state into the T state. So as protons are moving through ATP synthase, that energy is harnessed to drive a conformational state from the F1 through the FO in order to drive this L to T conformation. Once we're in the T state then so a free energy-driven conformational change converts L to T and catalyzes the formation of ATP. This step also involves a conformational change of the other 2 alpha-beta pairs. So as one alpha-beta pair is changing, so are the others, right? So L goes to T. What was in the open state becomes a loose state, and what was a T state becomes an open state. The reaction forming ATP is essentially at equilibrium under the enzyme's active site once you are in the T state. The free energy supplied by proton flow facilitates the release of ATP by converting the T to the O state. So as we enter into the O state, ATP is released, and as more protons are pumped, we can then reconvert the other alpha-beta pairs. So we go from the T to the L to T to O to release another molecule of ATP. So ATP synthase can be considered a rotary engine. So in order to have a rotary engine, you have to have 2 components, a rotor and a stator. The rotor is considered the C ring along with the gamma and epsilon subunits. The stator is the alpha beta, is the A and the B, as well as the alpha beta dimers. Protons from the inner membrane space enter into a hydrophobic channel between the A and the C ring, and then bind to an open C unit. The C ring rotates nearly a full turn until the subunit reaches a second hydrophilic channel between the A and the C that opens into the matrix. If ATP synthase generates 3 ATP per turn, and there are 10 C subunits, then the passage of each proton generates about 0.3 ATP. Rotation of the C ring. The interaction of a proton binding to a C subunit causes a conformational change which then causes it to mechanically push against the A subunit and is what drives the rotation. So this is actually driven by interactions between an aspartic residue on C and an arginine residue on A. So the electrostatic interaction is what drives the C unit to orient itself to that arginine 210 on A. However, as the proton comes in, it binds to that aspartic acid residue, neutralizing it, and that causes the C ring that then continue on rotating so that the next open C unit can interact with that arginine 210. So you can see how the protonation, the continuous protonation, causes the continuous rotation. So now that we've considered the function of ATP synthase and we see how protons drive the conformational change, which causes those alpha-beta subunits to bind ADP and PI in the loose state. Okay, so ADP plus PI are bound in the loose state. A proton is bound to the C ring, which causes a rotation. That rotation then causes that AB subunit to go to a T state, which then drives ATP synthesis. Another proton binds to an open C unit, and we get another turn, which drives us to the O state, which then releases ATP. Another proton binds, and that alpha-beta subunit is then converted to an L state so that it's open to receive another ADP and phosphate. Okay, so the one thing I want you to consider is if we get 2.5 ATP for every NADH and 1.5 ATP for every FADH2, how much ATP total can the complete oxidation of glucose produce? Now remember, this is under perfectly ideal conditions. Also, do not forget to add in the ATP generated from substrate level phosphorylation in both glycolysis and the citric acid cycles. So you may have to go back in your book to look at how much is produced in each of those. Okay? So we'll talk about that in class the next time we meet. So, obviously the coupling of the electron transport chain that produces that proton gradient and oxidative phosphorylation that harnesses the energy of that electrochemical gradient dissipation is important for ATP synthesis. However, as you remember in our case study that we talked about, we are able to uncouple those two processes if we can use uncoupling agents. So uncoupling agents such as DNP increases the permeability of protons across the membrane of the mitochondria. These agents uncouple oxidative phosphorylation from the electron transport chain by dissipation of the proton electrochemical gradient. So remember, DNP can carry protons across the membrane, which would then dissipate that proton gradient. Remember, one of the side effects we talked about when we uncouple ATP synthesis from the electron transport chain is that we're going to generate a lot of heat. So your textbook also discusses how this could be helpful in hibernating animals. And so it would be interesting, and I recommend reading over that subsection about how that happens. So the majority of the way that we control oxidative metabolism is basically through the citric acid cycle and glycolysis. Those are sort of our big points of control. The electron transport chain is controlled mostly through equilibrium, as most of the steps are near equilibrium. So if product's low or reactants are low, we're going to drive the electron transport chain through that. However, the mitochondria can regulate ATP synthase through the protein IF1. What's interesting is that in actively respiring mitochondria in which the pH of the matrix is high, IF1 is in an inactive tetramer form. However, if the pH drops below 6.5, the protein dissociates into dimers. And in the dimer form, it can then bind and inhibit ATP synthase. So thinking about that, can you predict or explain the interactions that hold the dimers together in a tetramer that is inactive? All right, so think about that. Think about what are some of the intermolecular interactions that exist in the tetramer that do not exist in the dimer. And we'll talk about that in class as well. So let's review our overall regulation of the catabolism of glycolysis. So if you remember, in glycolysis, we can regulate glucose, we can regulate hexokinase. We can also regulate phosphofructokinase as well as pyruvate kinase. We can also regulate pyruvate dehydrogenase. And then in the citric acid cycle, we can regulate citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and then a number of these other enzymes are also regulated by the levels of NADH, ATP, as well as some of the down-chain products such as citrate, acetyl-CoA, fructose 1,6-bisphosphate. So this would be a good time to go back and review all of these regulation points because this could be a good exam question. All right, so that's basically what I wanted to cover in this lecture. Obviously, we'll go more in depth and detail when we get into class and work on our workbook. If you have any questions, feel free to post on Facebook or email me. I'll be happy to answer any of them. And until then, I will see y'all in class. Have a great day, guys. Let's see.